So I think I'm moderating the next session up here, and we will get started right away here. So I am pleased to introduce Sally Aitken from UBC. She will be talking to us today on informing conservation decisions for making new climates with genetic data, lessons from widespread tree species.
Okay, can you see my screen okay?
Yes, we can.
Okay, so thanks very much for the invitation to speak here. And the talks so far have been so good. I'm just so impressed what people can do with small plants that have short lifespans and some really great experimental work as well as observations out there. I want to acknowledge that UBC is on the traditional, ancestral, and unceded territories of the Musqueam First Nation, and I'm coming to you from a small island called Quadra Island, which is the territory of the Lakwotek people, including the Wiwaikai Nation. And I'm very grateful that I get to hang out on their beautiful territory. So I'm going to talk about some things that we've learned and some questions that come up in our work with widespread tree species. I'm not going to go into a lot of detail on the methods we've used. I'll provide citations for those, but just give you an overview of our situation and maybe some things that we can apply to other species from this. So I work on mainly conifers, and conifers have very long, slow lives. And of course, they're facing rapid change. I'm always envious of people working on model systems because a species like Douglas fir is anything but a model system. We have the ability to maybe get through one or two generations in a scientist's career for a breeder that's doing crosses. And we have very large genomes, so about 20 gigabase genomes on average. And so these don't compare very well to things like E. coli and Arabidopsis or even Mimulus. About 34% of conifers globally are threatened, and I won't be talking about any of those threatened species today, but just to note that this is an ancient and not terribly species diverse group of organisms. They're not very species diverse, but they do dominate about a third of the world's forests. And climate change may be especially challenging for long-lived species like conifers, and especially if they are locally adapted. And most of our temperate and boreal conifers are locally adapted to climate. And here's an example from Sitka spruce. These trees have all been cut out of a common garden that we had at UBC. They're all the same age, planted in the same place. And above each tree, you can see the provenance of origin, ranging from California to Alaska and ranging from 12 degrees mean annual temperature to 3 degrees. We see that growth it shows a lot of local adaptation, and that largely reflects adaptation to low temperatures versus competitive growth in warmer environments. Now, in British Columbia, we've done a lot of work on local adaptation. And in fact, foresters have understood local adaptation in trees for centuries. But we see big changes happening in our forests, and we also have projections that show that there is a lot more to come. So up and down the west coast of North America, of course, we've seen a lot of increase in drought, in fire, in insect outbreaks, and even some disease outbreaks. These maps show the climatic niche projections for our 16 major ecosystem types in British Columbia. And we see shifts also in projections for tree species ranges. And so this is a middle-of-the-road projection, and if you compare the left mapped ecosystems from last century, second half of last century, to later this century, which is well within the lifespan of these trees, you see a wholesale redistribution of those ecosystem types. And we know that populations for many of these species are locally adapted, and we expect are becoming increasingly maladapted. So from a conservation standpoint, so much of our work has been motivated by informing reforestation. And in fact, assisted gene flow is well underway in the context of reforestation in forestry. But from a conservation standpoint, what do we really want to know? Well, we want to know if populations are locally adapted to climate. And we can't assume that all populations are, of course, for all species. We need to understand what climatic variables or other biotic or abiotic factors are driving local adaptation. With the species that we work on, we don't have a lot of local adaptation to things like soil, and climate is the overwhelming signature, but that's not true for a lot of other species. We need some idea about the predicted extent. We'd like to be able to predict the extent of maladaptation to future conditions so that we can understand if intervention is necessary. We'd like to have an idea of the capacity of populations to adapt to new conditions without intervention. And there's been a lot of talk about assisted gene flow at this symposium, which is awesome. Really great tests going on, like Nick presented earlier today. And we need to answer this question: is assisted gene flow indicated? And it won't be in all situations. And just Rebecca Jordan, Haley Tumas, and I put our thoughts on all of this in a conservation context into a recent review paper, if anyone's interested. So generally, most conifers are locally adapted to climate, but the extent of that local adaptation does vary with species. We've got species like western larch and whitebark pine, which does happen to be endangered, that have relatively weak local adaptation to climate. And then we've got species like Douglas fir and lodgepole pine that have stronger local adaptation. And just a reminder to everyone that species with small effective population sizes, small ranges, or high phenotypic plasticity are less likely to be locally adapted. So I think when we think about species that have been historically rare, species with small ranges, isolated populations, those are less likely to be locally adapted. And so we need to be very careful about how we think about moving things around in those situations. So this extent of local adaptation, we really need to understand on a species by species basis. And here's some data for the extent of population differentiation, which reflects local adaptation in Douglas fir. So Douglas fir has two major varieties. We've got the coastal variety here in green and the interior variety, part of the range, in purple. This is work of Rafael Candido Ribeiro for his PhD. And so for cold hardiness, we see typical relatively strong variation among populations that reflects local adaptation to low temperatures in those environments. So about 30 to 40% of the phenotypic variation is due to variation among populations. But when we look at drought tolerance in experimental treatments, we see that very little of the variation in coastal populations is due to variation among populations, and some of it, but still relatively weak among interior populations. So this extent of local adaptation depends not just on the species, but of course on the trait. And so we've looked at this in a number of species, and we keep finding relatively weak or no local adaptation to drought. We think that that could be due to a number of things. We have pretty good global adaptation to drought in these species, in some of these species. And we also have highly heterogeneous water availability across environments. So that may, so this very high level of spatial heterogeneity in water availability may make it very difficult for local adaptation given the high gene flow levels. So Douglas fir, relatively strong local adaptation to temperature-related climate, but we look at western larch. This is the PhD work of Beth Roskelley, and we see weak local adaptation for growth, phenology, cold hardiness, as well as drought tolerance. But western larch has a fairly small range. So these are some of the clines that Beth's found, but these are relatively weak compared to what we find for many of our other species. So there's been a lot of interest in using genomic data to inform us about local adaptation and to predict maladaptation in the future. And I know Brandon Lind gave a talk about this yesterday. Unfortunately, I had to teach at the time, but I'll build on what Brandon had to say a little bit here. So we really trust phenotypes, and phenotypic data is awesome. It is great for understanding what is going on. And so we can look at these data from lodgepole pine. This happens to be a test of cold injury in artificial freeze tests. And we've got 280 something populations here. And we see a strong pattern from low cold hardiness populations in the southwest to high cold hardiness populations in the northeast that corresponds very nicely to a climatic map of 30-year extreme minimum temperature. So we took a look at genomic data to see if we could find the same environmental drivers of local adaptation in genomic data as in phenotypic data. So we did exome capture on these materials, and we had phenotypic data from a 20-year height growth in a many-site provenance trial of lodgepole pine. And so what we did is looked at, compared 186 genotype environment association SNPs with, and the extent to which those SNPs predicted the climate that populations came from. We compared that to the climatic variance explained in this provenance trial for height growth. So basically, stronger climatic drivers should be located out to the right in terms of the provenance data and towards the top in terms of the genomic data. And we find a very nice correspondence between the two, where low temperature variables are showing up as being most predictive, most associated with population differentiation in both of these data sets. So things like mean coldest month temperature and the extent of continentality have very strong signals in both data sets. And that's a nice result, because then we can have confidence in using that if we're designing assisted gene flow. Interestingly, moisture variables came out weaker in the phenotypic data, but reasonably, moderately strong in the genomic data. So yesterday, Jill Anderson presented the really nice work on Buchra, putting in a variety of types of trials and showing just the strength of reciprocal transplants and provenance trials. And so with phenotypic data, of course, we can look at genetic clines in single environments for some trait. We can look at population responses across a bunch of environments, or we can look at relationships between how far you move populations and what effect that has on traits. And so These are all just great sources of data, and these are the types of data that have been used to design provenance trials. Somehow, all of a sudden, I'm writing on my screen, which is a little bit odd. Sorry about that. So these are great data for designing, reforestation strategies and that kind of thing. But if we're using genomic data, are we able to use those data to get the similar kinds of relationships? And here we have just a depiction of, we've got some phenotypic cline along some environmental gradient. And do we expect to see alleles following similar patterns, the alleles that are underlying adaptation? Do we expect to see localized genomic clines? And so we've taken a look at this. And so for lodgepole pine, for example, this shows clusters of SNPs that show similar spatial patterns for lodgepole pine that are associated with low temperatures, that is, with cold hardiness. And I wish I could clear the scribbles from my screen, but I'm not sure how to do that. So, so here you can see 6 clusters that all show different patterns for the frequency of warm associated alleles. So in pink, we have an allele that's at very low frequency along mean annual temperature until you get to quite warm temperatures. But in purple, we have a cluster of alleles that have changes in allele frequencies or show a cline along cold temperatures, but not much along warm temperatures. So quite a range of patterns here. And this really shows more of a localized pattern of clinal variation and suggests the complexity of these kinds of relationships. We find that that there's different genes and there's different SNP clusters that are providing adaptive diversity in different parts of the range. So again, these are the 282 populations that we genotyped, and we see variation. This is just for this cluster 6. We see that we've got relatively low allele frequencies in those clusters on the far side of the Rockies, and higher frequencies on the west side of the Rockies. But the good news coming from this is that we found that almost every population of lodgepole pine that we genotyped was polymorphic for temperature-associated SNPs. So there's very high standing variation. The warm alleles are present even in most of the cold populations, and the cold alleles are present in most of the warm populations. And that bodes well for long-term capacity for adaptation. Now, that was a reasonably clean story in lodgepole pine, but Rafael Candido Ribeiro has been doing some of the same things in Douglas fir. And so he's looked at similar clines, first of all, for alleles associated with drought tolerance from his GWAS experiment. This is the interior variety of Douglas fir. This is the coastal variety. And so this is the frequency of alleles increasing drought tolerance. These were identified within population in GWAS. And you can see that, especially in the coastal variety, we've got some alleles going up in drier environments, as you would expect, but we've got other alleles that are decreasing in frequency when you would expect them to be increasing in frequency. So a great deal of complexity. And even when we look at cold, which is usually our go-to trait showing the strongest population differentiation and clinal patterns, we still see some clusters of SNPs that are going in the opposite direction we would expect. So we see these complex clinal patterns that are much harder to interpret than the phenotypes. And we think that there's a lot of genomic redundancy going on here, where you can get the same phenotype with different combinations of alleles. And so you don't find these general consistent global patterns of clines. But I'm sure Brandon talked yesterday about how genotype environment associations are much more complex than we ever anticipated, and that the patterns that we find will first of all will only see significant declines for a fraction of the genes that underline variation in a trait, and that can arise through genomic redundancy, polygenicity, pleiotropy, and also that you've got multiple environmental gradients underlying these patterns. And so this is a paper by Katie Lauderhouse, who's along with Brandon has done, and others, has done a lot of great work in teasing apart through simulations the complexity of the patterns that you might see. So this kind of bursts one hope that I had, which was that we could use genomic data to get information on local adaptation that would be of similar quality to what we get from phenotypes, and that would let us design things like assisted gene flow. I still think there's a lot of information in the genomic data. We shouldn't disregard that, but we have to be careful about how we interpret it. And I'm just going to show you one other thing from that very quickly. Predicting genomic offsets has become a big thing with genomic data. This is predicting maladaptation into the future. So first of all, we're identifying markers associated with climates or climate-related traits. We're predicting the genetic environment relationships over large areas, incorporating climate change predictions, and then validating those predictions so that we can hopefully say what populations are going to be most maladapted in the future. Now, this approach can be quite useful for identifying differently adapted populations. This was an effort to identify seed zones using gradient forests, and it consistently identified the top climate variables from a set of unselected SNPs and also from a set of candidate SNPs. The results were very similar. So that was quite encouraging. The genomic data can be used to cluster similarly adapted populations and to identify these the climatic drivers. But work that Brandon did to predict maladaptation in Douglas fir and to validate that work that I think he mentioned yesterday brought up some good results in terms of identifying climate variables and performing better than climate or geographic variables alone. But if we look at predictions of maladaptation here, species-wide across two varieties versus just for the coastal and just for the interior variety of Douglas fir, what we find is very different predictions depending on what the training populations are going into this, where this set of populations was predicted to have very high maladaptation in the future based on this model, and low risk of maladaptation based on the interior variety model only. So lots of caution in extending these models too far. So I know my time is up. So I just want to say that we can use phenotype or genotype environment associations to identify climate drivers of local adaptation. But the genomic signals of local adaptation are really complex. And these predicted genomic offsets are not yet proven for making management decisions, in my opinion. With trees, we've got to be careful we don't move genotypes too far too fast. They are going to get cold injured, or they have the risk of getting cold injured. And we really need to be cautious around certain types of transfers, especially elevational transfers are riskier than latitudinal transfers. We may want to mix populations, that is, do more composite provenancing. We don't have risks of outbreeding depression in our situation to any extent. And we know that from many long-term tests, but that may be a risk in some systems. And of course, for many of you, the potential for adaptation to non-climatic factors should be evaluated, as well as outbreeding depression. That was a rush through a lot of stuff. These are the people who have done all the work, and I am happy later on to discuss any of this. Thank you.